Oh, okay. Got it.
Thanks for joining us today, Lifan. We're very excited to hear your work. As you know, we're all interested in how different groups study large collections of iPS cells and their differentiated derivatives. And your study really is kind of an extreme version of that, using lots of lines and doing quite an interesting readout in the sort of the neuronal activation transcriptome. Especially in the context of complex brain disease genetics. Very interested to hear your work, and thanks for joining us. Take it away. We can see your slides just fine.
Okay. Thank you. Hi. So I'm Lifan. Here to present our project about is a single project from Xin He and Jubao Duan. So let's start. How to? Uh, yep. So first, a background. We have done like a GWAS for neuropsychiatric disorders for many years. Here is like a diagram about 2022 schizophrenia GWAS, all the different signals, about 100 loci. Um, and we also try to use like EQTL to explain how these risk loci convert their risk into these molecular traits and then to the phenotype. However, the studies have shown that uh, despite like our efforts, usually only like 10% of those GWAS risk signals are actually mediated by the EQTLs. So possible reason for that is the existing resources like a GTAS or a Cycling they usually rely on a steady state or postmortem tissues. And the activity-dependent genetic regulation is usually masked in these sort of steady state samples or bulk samples. So how does the activity-dependent EQTL relevant? So that has already been like verified from another different area, immune cell activation. Multiple studies have shown that EQTLs are only identifiable in immune responses. They were enriched in immune or autoimmune traits. But in the neuron stimulation context, usually there have been very rarely studied. So here the hypothesis about this project is basically presume that many risk variants for the neuropsychiatric disorders, they act specifically during the neuron stimulation states, which is important for plasticity and usually not captured in like a standard postmortem profiles. So here is like the overall study design for the project. We started with about 100 cell lines for the human iPSCs. And we used well-established protocols, basically, from like 2013 or 2017 protocols that basically use the lentivirus to force the TF expression and then basically induce like a pure population of glutamatergic cells or GABAergic cells. And because it's like pretty fast and reliable, well, there are still some differentiation failures, but relatively small. And after the differentiation, we pull these separately cultured neurons together with the rat astrocytes into a co-culture system for around 4 weeks to like make the development of like a biological neural network. And then after these 4 weeks, we basically separate into three different conditions. One, for control, we just put at rest for 6 hours. For a short-term response, they're put at rest for 5 hours and stimulated like 1 hour before the harvest. For the long-term response, we basically stimulated with the KCL like at the very start. Uh, well, because like there are supposedly may be some rhythmic expression patterns. So we are doing this to make sure all these different conditions they were harvested like at exactly the same time point to reduce confoundings that could be due to like a cycling expression patterns from like a somewhat biological neural networks. So after the harvesting, we got the single nuclei, single cell multiomics. We check the canonical markers and separate them into the GABAergic and glutamatergic cells, and also subtypes. And we run like EQTL and CQTL mapping. We also check like which sort of QTLs there are only evidence in the stimulating states. And then we also like run all these QTLs we found from all different contexts and see how they're associated with neural brain disorders. So I need to mention that we also run the GRN, gene regulatory networks, comparing like 18 cell lines before and after stimulation. That part is not really covered here. It was done by Zicheng. So here, first of all, we check the markers.
Can I just
And
ask you a question about that
okay,
slide, just so I'm clear about it? So
sure.
on the left-hand side here, you have 100 lines in total. But
Yes,
did you study the lines one by one, or were they pooled?
they were pulled. So, well, I don't get the details. So this part I feel like is part individually cultured, but in the co-culture system, they were pulled. So the pool is like 3 to 5 different cell lines together. So each co-culture batch will have 3 to 5 different cell lines.
I see.
Yes.
I see. Thanks very much. Yeah.
Since Ron asked a question, can I ask one too? So how did you sort out for single cell RNA sequencing the rat astrocytes versus the human cells? Or were the rat astrocytes also sequenced?
That's a good question. I think probably they are also together sequenced, and then from the aligned reads, we remove the reds that are aligned to the reference genome of rats.
Yeah. Actually, we know the answer to that because we did something similar, a very simple version of this a long time ago. You can easily distinguish the rat from the rat by the sequence. Isn't that fair, guys? Yeah, you know better than I.
Yeah.
That makes a lot of sense. So did that reduce the overall total number of cells? So like approximately how many cells per sample were you able to get? Like human cells per donor per
That's a
condition?
good question. So yeah, that will induce like somewhat of a waste. So if I remember correctly, GBAO's lab, when they do the co-culture system, the proportion is around like 5 to 1. So basically for each like one astrocyte cell, there are like 10 neuron cells. They are either glutamatergic or GABAergic.
Gotcha.
So when we remove the cells, it's not like a high percentage. Actually, when we do the QC, I feel like we remove like a 30 to 40% of cells from the single nuclei. Like so, the part that the waste caused by the rat exercise, I would say, is like a not like a very major factor.
Okay.
I
And sorry,
go for it.
do you remember how many, like approximately how many cells per donor, just for EQTL analysis? Like, did you feel like you had enough cells and reads per cell to be able to call QTL? As clearly you were able to call QTLs, but um,
Yeah, the cells actually, number of cells vary a lot. I'll mention that lately a little. So we need to do a more stringent QC. So yeah, we do different things. Sometimes we try to remove it in the by the read counts. Sometimes we remove some samples based on the cell counts. And also the risk per cell is quite different too. What we finally did is like I think it's a total risk we got from each donor, regardless of the cell count. Well, the cells also get QC, but yeah. And so although we have like 100 cell lines, we remove at least 5 cell lines from the QTL mapping. For some other application or yeah, EQTL, different contexts, we also do a different QC because the cell type, some cell type has more cells, others has little.
If it's okay, Lifan, I have one other question for you here while we're talking about the experimental details.
Oh,
So,
okay.
and it's actually from something you mentioned that I hadn't thought about it. You have the 1 hour and 6 hour stimulations in which the culture is really going to be screaming, right, from KCL. But then you also mentioned that in the control situation, while there's no stimulation, these are neurons that have been creating networks for a good time. I think it's about looks like
4 days
28
now.
days. Yeah.
Yeah, yeah.
So did you look or do you have an idea of how much spontaneous activity is going on in these dishes without stimulation?
Uh, yeah, I think Zhibao's lab, they have provided some fluorescent assay to show that neuron activity, like when
Using
you're not
calcium?
stimulated. And, yeah, yeah.
Okay,
But I'm not sure what the fluorescence is about. But they all have those assay.
cool. Thanks.
Okay, we'll move on to. So we basically separated into the cells into like a glutamatergic and GABAergic. But when we look at the UMAP visualization of the RNA-seq, there seems like 2 different patterns within the glutamatergic cells, and they can be divided by the NEFM. So basically, it's one part of the glutamatergic cells, they have a higher NEFM expression than others. So we divided all these neuron cells into actually 3 different subtypes: GABAergic, NEFM positive, glutamatergic, and then if missing glutamatergic, and we discarded other ambiguous neuron cells. And then we transfer the cell type label to the ataxic we got and run the UMAP visualization. It seems like the pattern is still consistent, like basically 3 different neuron subtypes. So we also compare our neuron cells because to another study that has actually sequenced the cortical samples from different development stage of humans. And we compare the expression and try to predict which development stage are our cells in. And here is showing that from the GABAergic or glutamatergic cells, predominantly we have our neurons are mapped to the second trimester, like at least half of them. And then maybe some of them are 2 to 1 or 1 to 2, and others are basically a very small proportion. And we also check the cell type composition. So here, this is each column is a cell type composition for each cell line. So we have like 100 column. Basically, this showcases the composition across different cell lines. And each panel is a different time point. So you can see that from each cell line, actually the cell type composition varies a lot. But if you see, we observe like different time points together. Actually, the pattern is quite consistent. So we would like to say that like the neuron stimulation didn't induce any sort of like cell type specific deaths or selection at least. So I would say like when we compare the stimulation to the stimulating stage to the resting state, it would be like a fair comparison to interrogate the stimulating specific gene programs or QTLs or other stuff. So here is the trajectory analysis run by Zicheng, another co-first author. He run another pseudo-time computation analysis to verify that and derive like early response and repression, late response gene modules. It's basically like the first one third will correspond to the resting state, middle one third will correspond to the 1 hour, and the later one third will correspond to the 6 hours. And as you can see here, basically the trajectory in different clusters, the average gene expression will be somewhat stepwise, meaning like within the same time point is very similar. So we say like they each cell type in each time point, they were quite homogeneous. So when I compare them using a pseudo box sample approach, I would say it's somewhat reasonable. And so here I just directly show the results from our EQTL mapping and the CQTL mapping. We use all sorts of covariates like cell type proportions, age, and sex, and others. And then we derive these genetic effects for each context. So first of all, I need to say like we have the three different cell types, and within each cell type, we have different time points. Basically, resting state 3 hour, early response 1 hour, late response 6 hours. So together we have 9 different cellular contests. And we run EQTL mapping for cells within each contest. And here is like the number of genes with at least one significant EQTLs. And you can see here, like compared to the resting states, mostly we have like several hundred genes that can only find these genetic effects in the stimulating states. And this phenomenon is more obvious when we look at the chromatin accessibility QTLs. So basically here is showing that like several thousand peaks, they can only find like the genetic effects regulating this accessibility. They only show them in stimulating states in the early response or late response. And we also compare our EQTLs to another study that has run dopaminergic neurons and with very different development stages. I think what they did is like they have different cell types, dopaminergic, serotonin, progenitors, and different stages at day 11, 13, 52. And they also have different treatments. We compare our EQTLs from the 9 contests to all the different stages and treatments from their study. And what we found is like we match basically mostly with the day 52 dopaminergic neurons, or maybe some also from the day 30 neurons. And then we try to identify EQTLs that actually only show themselves in the stimulating states. So what we do is like we run a very standard interaction test. Basically, we see the coefficient for the interaction between genotype and time point, whether this is significant. And here is data showing like how many genes we found from the tested genes. Well, although the proportion is unexpectedly high, if we look at the proportion of the EQTLs, actually is like less than 1%. So basically all the significant genes we found here, for each gene there may be like one, usually just one dynamic EQTLs. So here is one example of the dynamic EQTLs that we found for the ADAM10. Basically, 0 hour and 6 hour, when we do like a EQTL mapping marginally for this contest, we did not see any EQTL effects. But when we do the EQTL mapping with the 1 hour within the MP group cell type, we can see that the dynamic effects is actually quite significant, and the direction is somewhat different from the other 2 time points. And we also try to compare the dynamic EQTLs with the bulk samples from the GTAC Spring EQTLs. As you can see here, if we directly compare the resting states of our EQTLs, around like 60% or higher proportion of them will be significant in the GTAC Spring EQTLs across different tissues. But if we use our dynamic EQTLs and test how many proportion of them will be significant, only like 40 or 20 or sometimes close to 0. So the percentage that are significant in the GTACs would be much lower. So we would say like the dynamic EQTLs tend to be like missed by like both sample atlases. And so this like we have one plausible reason why the EQTL would be dynamic is that there are chromatin regions that are only open in the stimulating states. And because the dynamic EQTLs, these genetic effects only located in the upregulated peaks in these stimulating time points. So that's why these EQTLs only show effects in the stimulating states. So to verify that hypothesis, we basically run an enrichment analysis with the EQTLs against the upregulated peaks in the stimulating states. What we found is that basically the dynamic EQTLs are like, I would say that 10 volts to 20 volts around, more likely to be located in the upregulated peaks compared to the background SNPs. So now we kind of established that we can find dynamic SNPs, and they're maybe missed by the bulk atlases, and they may be driven by the upregulated peaks in the stimulating states. But like, are these disease relevant? So to answer that question, we're trying to associate our EQTLs to the GWAS SNPs, trying to explain like whether the dynamic EQTLs or context-specific EQTLs actually mediates the GWAS signals in neuropsychiatric disorders. However, there's some caveats. So basically, this is there's a scenarios that it would cause like false positive risk genes because of the LD in the GWAS SNPs. So the SNPs in the LD, it could also have EQTLs here. But this gene, the X, it doesn't really have a causal link to the traits. But due to the LD and the EQTL we found, we may draw the conclusions that this gene is actually a risk gene. So Shin's lab developed a method called the causal TWAS. Basically, what they do is doing fine mapping across the whole GWAS loci, pulling all these GWAS SNPs, also with the predicted gene expression all together to run the fine mapping. So let all these risk SNPs and the risk genes, candidate risk genes, compete for the risk signals and figure out which one may be actually like a putative causal. So what we did here is one step further. Basically, we pull all the EQTLs from all different contexts and to run this and let them compete for the risk signals from the GWAS. And the heat map here shows like where the evidence source come from. So for example, like CPT1C here, only one contest from the 1 hour NM groups that actually provide an EQTL explains that can mediate the causal signal in that loci. On the contrary, if we look at the MPIPP2 here, basically it shows like you can find EQTL from various contexts, and they will all mediate the GWAS signals. Similar thing is the SERP1N1 here, which these two are like well-known restraints that are already discovered by using the GTAC spring tissues to mediate the GWAS. So what we found is that with our dynamic EQTL or context-specific EQTLs, we found around 48 restraints. If we use, most of them are not really found if we use the GTAC EQTLs to mediate these GWAS signals. Here is one example of the restraints we found, the CPT1C. So within that GWAS loci, if we only run the T1s, basically we may conclude that PRRG2 and CPT1C, both of them are restraints, and PRRG2 may also like confer the risk to GABAergic cell in different cell types. However, if we run the fine mapping with all these restraints in a different context together, what we found is that only the CPT1C actually explain the causal signals. And this is a gene, it's around the enhancer region. So CPT1C is involved in the metabolism of branching fatty acids and predominantly expressed in neurons. We hypothesize that lipid profiles may increase risk for the schizophrenia. We also run the part in the CAQTLs too. And what's interesting is that CAQTL actually mediates much more signals than EQTLs, as shown here. It's like 2 times or 3 times more across these 5 different new MPD traits. So here's another example from the CAQTL. We have different sources, well, not just CAQTL actually. We have the CAQTLs of the risk peaks, and they also have the allelic-specific overchromatin. So by using T1s, we may find that our different peaks, regulatory elements, they may have an effect on the schizophrenia. However, running the fine mapping with the causal T1s, what we found is like this one peak and this allelic-specific overchromatin, they may mediate the GWAS signals, and they're located at the MET1L1. However, we didn't really have the EQTL here for the MET1L1. So what we actually found is just a signal of the regulatory elements that can mediate the GWAS risk for the schizophrenia. And when we actually try to locate the risk peaks for the risk genes, actually only 2 of them are like somewhat closely co-located on the CNF and the CYMYD. Most of the signals from the risk peaks, they're actually independent from the EQTL signals that we can find. So for the summary statement, a substantial proportion of genetic effects we found like regulating the gene expression or the chromatin accessibility. They're only identifiable upon the neuron stimulation. And dynamic EQTLs are likely missed by the existing steady state atlas. Also, these EQTLs were enriched for the GWAS signals, and CAQTLs may mediate much more heritabilities, and they're independent from the EQTLs we found. So we're still looking into downstream activity from those CAQTLs. Again, this is a project of teamwork. A lot of people is involved, and thank you for a lot of lab members from the Shins and Chibao's lab. So for the multi-causal CT1 software is please check out this web page. And our work has been published in the Science lately. Also, please read the manuscript to figure them out. There are a lot more details I didn't really cover in these slides. And yeah, that's basically it. Thank you.
Thank you. And please, everybody, jump in with questions. Looks like Shiraz, you have your hand up. Go ahead.
Yeah, thank you so much for a really wonderful presentation. I wrote down a few questions for you, actually. So I guess the slide where you talked about your cell type proportion distribution by samples, right? I'm just curious to know that when you're tagging your cells, did you come across a high amount of overlapping, and how did you deal with that if you did?
High amount of overlapping for cell tagging.
For cell types, right? So like were there cases where you had ambiguous identities and how did you resolve that?
Actually, we just discarded that, I think. So basically here, the satellite proportion here, we basically, what we use for the downstream analysis, the green, the brown, the purple. And for the glutamate they are ambiguous, or sometimes they are actually unidentified. We didn't really use that for downstream analysis.
Okay. Okay. Thank you. And then I guess another question was about the Pi1 analysis you ran with GTEx. So I think you made a point of how the profiles of your neurons are largely associated with the gestation phase. So then the question is, how relevant actually is the Pi1 analysis with GTEx, because GTEx is primarily composed of adult postmortem samples, right?
Yes. Right. So basically, the point we're trying to make is, well, first of all, we need to validate whether we are consistent. And GTEx may not be we are consistent with the current neuron cell findings. While GTEx may not be a very appropriate reference, because like as you mentioned, these adult postmortem bulk samples, still we can find some association. Well, actually, it's higher than we expected. Actually, I thought like I would expect like 40% significant or something, and then it goes to like 60. And we also, so that's why we actually compare with a lot of other EQTL addresses like the Gerbers or the ROSMAP and others. They were in the manuscript. So yeah, but then we specifically try to, because it's like bulk and postmortem, we would expect like the dynamic QTLs are even less present in these samples. So that's why we chose for like a comparison here.
Got it.
Yeah.
And sorry, one last question, if I can get one in, is would it make sense to do any colloc analysis as a follow-up to the fine mapping?
Colloc analysis. So you're saying like after the fine mapping, we check whether the EQTL actually colloquialize with the GWAS receipts?
Yes.
Let me think. Could be reasonable, but TWAS actually is like a somewhat like a colloquialization, except that it's running like it implies the MR frameworks by doing a prediction. So basically, the EQTL, we use the EQTL to predict the gene expression. The EQTL is already has the p-values from the GWAS. So when we try to impute the z-score, like what we show here, so the p-values for these risk genes, they already somewhat colloquialize because the SNP that support the EQTL, they need to have a p-values from the GWAS. And that GWAS p-values will convert to the gene by the EQTLs. So it's implicitly colloq analysis already. What we do is like we use all these, you can think of this like plausible genes that we found from the colloq or TWAS, and we pull them together for another fine mapping, let them compete the same causal signal together, and then derive the one that could be like in the credible set that actually may be causal, putatively causal.
Interesting. Okay. Thank you very much.
Thank you.
We have two more hands up. May, you want to jump in first?
Sure. Hi, thank you for the interesting talk. My first question is, can we go back to the bar graph where you show 100 individuals?
Bar graph?
Yes, the stacked bar graph where you show, yeah, two more,
This one?
yeah, this one. So,
Oh,
no,
this one?
yes, this one. So,
Oh, okay.
here you're showing the percentages, but do you see any differences in the absolute number of cells? Because in this series of seminars that we've been watching, a lot of the time, what will happen is if you mix a lot of cell lines, a small number of cell lines will dominate the culture, and that's been, kind of a theme in this series. So I'm wondering, do you see any disproportionate growth of a small number of cell lines, or are they pretty much even across the culture?
Oh, wow. This one, I may need to defer that to Jibao's lab, but
Maybe
so
I can
for
help here, Lifan. You, at
okay.
least conceptually, you mentioned that the lines are not mixed until they are post-mitotic neurons. Is that correct?
Yeah, they are
So that
individually
I'm guessing
differentiated.
May would,
Okay.
right? So they're all grown individually,
Yeah. Yeah.
mixed after post-mitotic. So I'm guessing there's probably some variation, but not due to proliferation in the dish. Yeah.
Yeah. Thank you.
And Jason, yeah, jump on in.
Yeah, Lifan, so just on this slide, since we're on there, did you find, so were all this may be a Jubao question, but were all the iPSC lines generated in the same way? Meaning like, were they generated from fibroblasts or from blood? And do you see any differences in differentiation based on, the sort of a technical artifact of how the iPSC lines were generated?
Wow, that's a great question. I never thought of that. Um, so I'm not really sure. What I remember is like the cell lines are collected from multiple different ways. Some are bought from companies, some are like other. And when we check the genotype PC or others, yeah, it seems like somewhat heterogeneous in two different parts. Actually, you can kind of see the ID for maybe 40 cell lines. They are very different from the 60 cell line I got here. So yeah, there is some source heterogeneity, but I'm not really sure like whether like which tissue they're derived. I think Jibao once mentioned that they are derived from the whole blood or something.
Okay.
Yeah, but there are some heterogeneity, especially this part and this part will have a little bit like a genotype diversity involved.
Oh, so
Yeah.
they're from multiple ancestries as well? Like
They
the
are from
components?
Europeans ancestry, all of them.
Oh, they're from, okay. Sounds good.
Yeah.
And another question too, so you showed that you got CAQTLs and more CAQTLs in the stimulated condition.
Okay.
Were did you run any kind of like motif breaker type of analyses to know what type of like if the SNPs altered the motifs for any known transcription factors? Does that make sense?
Yeah. Let me think. We try to pursue the direction actually, and actually there's uh, one thing that has already been verified. The CNF823 here, the one that overlap. This one is pretty famous actually, and we can actually find that the CPICs here. Well, it's not dynamic, but the CPICs here they actually break the motif from the transcription factor and then disrupt the CNF823, and this was already reported by another study independently. But for the dynamic CQDLs, I haven't really pursued the direction and trace like like how the dynamic CQDL has like disrupted the motifs. What we did is like we're using more traditional GIN construction. That was done by Zhicheng. What I think is like they compare the differential accessibility piece and show like this piece where there are some TF motifs involved and then construct a GRN and things like that. But it's not really from the CQTL perspective.
Cool. Thanks. Yeah, I'd just be interested, especially if they're like immediate early genes, like those genes that are sort of known to be activated after neuronal activity, and to see if those would be particularly disrupted would be pretty interesting. Okay, thank you.
Any other questions?
Well, I was just thinking it might be worth just picking up on the point that we're discussing now. So, the FOS gene is an immediate early gene, which was very early on shown by Mike Greenberg and Hilmar Badding and Morgan Sheng, the sort of gurus of activity-dependent gene expression in their subsequent work, to be an immediate early gene in this context. And what I find very interesting listening to you talking is that for example, that image you show where you show that the GTEx EQTLs and the dynamic loci or signatures that you've defined here seem to be separate. It's as if what you've done, and it's like it's cool that you've done this, is you've sort of driven a whole lot of cells with the Wernig-Sudhoff directed differentiation strategy into a particular state where then you can look at the response to KCL. And let's see if I can pull this thought together. So what I find myself thinking about is that the whole of the chromatin is responding. And that's kind of what you've opened up here. That it's true, there's all kinds of specificity going on, but it's going on in this context where depolarization of these cells leads to a chromatin-wide change that you're exploring. I don't know whether people find that helpful, but I do, because it's something that hasn't really been done. And then you find yourself thinking about the timing effects and whether or not these QTLs that you're looking at here are actually regulating the precise point in a sequence of change following activity-dependent calcium inflow. Just to put one more sentence in here. So it's a computer, right? You have a series of genes being turned on by these events, right, in a particular sequence, and it's chromatin-wide. It's a very sort of cool world, it seems to me, you're opening up in front of us. And there's quite a lot of work in sort of looking at activity-dependent events that's relevant to this. But I'm just trying to put your work in a context where maybe everybody can sort of think about it. It's a very, you might say this is what a neuron is about. It's about changing its patterns of gene expression in a precise way following activation. Anyway, thanks a lot.
Well, thank you. FOS gene you mentioned is that's like a very famous early gene. Actually, we also observed that, I think. I didn't really mention it. We observed the early response gene FOS or early gene B. FOS is actually interesting in terms of the chromatin accessibility thing. I think lab members has observed things like it has the enhancer priming, if I remember correctly. So it's also like cell type specific. So for the NFEM positive groups, the chromatin actually open at the resting state already. But for NM groups or GABAergic, or maybe a different. Well, for the other two cell types, the chromatin only like open exactly at the early response, like 1 hour.
Yeah.
So we
Yeah,
observe the
sure.
chromatin together with the gene expression spike up. But for the NP group, it's a little different. It's actually the chromatin open at the resting state already somehow, and the gene will still spike up. So if you only look at the gene expression, it's kind of the same behavior. The false gene just go up no matter what. But if we look at the chromatin accessibility, it's actually the behavior is different between these particular one cell type NP groups. Yeah.
Yes, you show that in the paper. Actually, it's very nice, and it adds a sort of another kind of wrinkle to this, that you have to understand the primed state, where the cell is coming from. And I mean, if people are still following all of this, BDNF, which is another classic activity-driven neuronal gene, is not an early gene. It's in the later phase. And so, you know, then you describe, maybe it's in one of the slides that's up on the board here, but you described a particular gene, a fatty acid gene that you focused in on with your fine mapping. And I wondered whether in that case you'd thought about, what is the protein regulated with the same time course?
Wow. Yeah, that could be true. I'm not sure.
Yeah, it's interesting. So for BDNF, for example, we know quite a bit about where the protein and the message are, you know, what their quantitative values are. Anyway, thanks. Very interesting.
Thank you.
Thanks so much, Lifan.
Oh, thank you. So we can end the session, or?
Yeah, I think if there are no other questions, thanks, and we'll catch you next time, hopefully.
Okay. Well, thank you. Thank you for all the great
Likewise,
questions.
thanks.
Yep.